On a more serious note, we're thinking about what we're going to talk about today, and we really wanted to talk a little bit about machine learning and AI, but we thought we'd center it around what do we get the most business value from. In any project we've ever worked on, we need labeled data, whether it's AI or if it's a customer client request. And our biggest gain that we've had and really helped us scale in Diaceutics over the years is really taking a step back and looking at our underlying data sources and cleaning it from the bottom up, really labeling that data. So, a little bit boring, but just a little bit about the company, I guess. At Diaceutics, we believe that every patient should have access to the right treatment at the right time, and we're committed to solving the commercialization challenges along the way to make that happen. It's really started from a vision from our current CEO, Peter Keeling, way back 20 years ago. Peter had vast knowledge of the pharmaceutical industry, worked for many of the top pharma companies, and really believed that precision medicine was the way forward. He disagreed and didn't really like the traditional drugs that were out there, especially in oncology, where likes of chemotherapy can be quite invasive and can be really destructive at times. And when the first precision medicine drugs started to come around, Peter thought, you know what, I'm going to devote the rest of my time and build a company that really focuses on this. So precision medicine has been our focus right from the start. Really over the last decade, with the age of data science and machine learning and everything else, data has become more available, especially in the US. So we have really concentrated on building our data source, our data lake up from many different sources, which I'll talk about soon, which gave us an unrivaled data set compared to any of our competitors out there at the moment. We have a global network of our pharmaceutical experts covering 53 different countries, and we're an end-to-end solution provider. Again, we're concentrated on the commercialization of precision medicine. So again, just a little bit about what we do. We work with and trusted by 39 of the world's leading pharma companies. So Pfizer, AstraZeneca, we have all strong relationships with those top pharma companies. We have a field-based team of 161 experts across 53 different countries. And we're really focused on disease areas, mainly oncology at the moment, but we're also diversifying outside of that. A little bit about our data lake. We've been building this up over many years. If anybody has ever worked in healthcare data or with healthcare data before, they'll know how siloed the data is. It's really broken up into various different components. So we have medical insurance claims, heavily in the US, whereby we can see how patients are being treated, how they're being tested, are they following NCCN guidelines. We have EHR data, which looks at the physician's notes, the encounters with the physicians and the doctors themselves. We have lab result data, which is very important. This gives us the genomic data, which tells us if patients are eligible to go on life-saving drugs. And last but not least is our pharmacy data, where we actually see what drugs patients are being received, whether it's a chemotherapy drug or a precision medicine drug. And what are we looking for in this vast data set, I guess? And this is it. This is what we call a patient pathway. We want to see everything that happens, how a patient's being treated, right from the very first time they see a doctor, right through the pre-diagnosis, the doctor gives that diagnosis, all those encounters. We want to see the diagnostic data, the genomic data, any blood tests involved there. And then it goes through to the claims side of things, where the patients are being diagnosed as ICD codes basically relating to the diagnosis of that patient. And we'll hear us talking a little bit about this as we go through. And that's why we hoover up as much data as we can. We follow HIPAA legislation, GDPR. There's a lot of legalities around that, I guess. But we really want to open up the patient pathway here so we can really truly tell our pharma clients what is happening in the field, how patients are being treated today, what the gaps are, and what they need to concentrate on. So around this, we have built many data products. our data products are really based around entities in our data. So we'll have insights based around our physicians. We know how physicians are testing. We know how they're treating, and we know how to educate, or which physicians to educate to make sure that they are optimal in their drug selection. We can see, again, the patient journey, the life cycle of the patients, if they're going into remissions, if drugs are working in the market today, or if they aren't. One of our flagship products today is called Signals. It's been released in the US. We are trying to move outside the US. It's just difficult with the legalities around that. But today, we offer a weekly or daily data feed to our pharma clients, whereby every week or every day, we select patients who are eligible to go on drug, who could potentially save their lives. And that's based on their genomic data, which we receive from our lab network itself. So I guess no matter what product we have and what project we work on with the client, if it's a more custom project, what we're looking for is a certain type of patient. Precision medicine is really focused on the type of patient they're after. They may want patients who have already received chemotherapy and maybe relapsed, or they may be looking for a genomic signature in the data. So it's very important that we're able to select those patients, but not only that, scale that as well. And this is really where Knime has helped us over the years. So without any further ado, I'm going to hand you over to Wendy. Wendy is going to talk a little bit about how we've utilized Knime, I guess, to identify clinically relevant patients and cohorts within our data.
Okay, so I'm going to talk about our rule sets, and this is really how we get additional value out of our data. So Scott showed you the patient journey, and we can see everything within that, but healthcare data itself is transactional, and in its raw form doesn't really give us very much insight. The journey that Scott showed, everything in our claims is coded. So we have ICD-10 codes for disease, we have HCPCS and CPT codes for procedures, and we have various drug codes as well. The ICD-10 codes themselves are top level only as well. So what that will tell us is that someone has lung cancer. What it won't tell us is that what stage of disease are those patients at? Are they early stage or advanced? And that's really where our clients want to get their data insights from. So that's really why we have focused on generating our labeling system. So the labeling system really helps us identify these patient cohorts. It also allows us internally for all of our analysts to be singing off the same hymn sheet, so everyone is using the same definition of disease. It speeds up all of our processes, and we can generate insights really quickly. And overall, everything's reusable, validated, and we have quality control around that. So I've been working in translational oncology probably over 20 years now, and whenever I first came into this area, back in around the year 2000, patients were treated as one big population. So an advanced lung cancer patient, for example, they were given a standard chemotherapy, and basically, if they didn't respond to that chemotherapy, they were moved on to the next chemotherapy. So the first chemotherapy was one that worked in most of the patients. Second line was the second best, and the third, and so on. The problem with getting treatments that don't work for a patient are that they lead to toxicity for the patient. It also leads to time wasted for those patients getting treatments that do not work for them. And finally, treating with chemotherapy drugs that don't work for the patient can actually induce further genomic changes within the patient's tumour that can be detrimental. So fast forward on to around about 2007, and we got publication of the breast cancer subtypes. And for the first time, we see that it's not one big population of patients, but they identified 4 subtypes. One of them was a fairly big one at nearly 70%, 2 at 10%, and 1 at 4%. And what was interesting here was that each of those subtypes only responded to particular treatments. So the treatments that worked in subtype 1 would not work in subtype 2. So that was our first sight of that personalization or precision medicine. Moving on to where we are now, there's more and more biomarkers and drugs being approved every day, and we're now getting down to maybe a 1 to 2% population, or even in some cases with rare mutations in a less than 1% of the population. So it's really important that we're not looking at the big population, that we can actually identify these cohorts. So just to explain that a bit further, within our claims then, what we can see within claims is lung cancer. Our clients are really interested in really delving into that data, looking at things like a patient journey, the testing rates, what's happening with those patients. And on the whole, that would be okay, but if we look at an early stage population versus an advanced stage population, the patient journeys are very different. They will get different drugs, different testing at different times. So having our labeling system there will enable us to actually get to that early stage cohort for our patients or the advanced stage. So it's really important that we do have the labeling system. So moving on to a couple of case studies that we have. So we have many different labels. We label as much as we can for our patients. Everything from stage of disease through to line of therapy, subtype, as many labels as we can generate, we do. And I'm going to show some that we have done on advanced or stage of disease, so advanced lung cancer cohort. So this is one of our Knime flows. So we've utilized Knime in terms of all of our labeling. And what we are able to do is this process here. So it's a repeatable process across all solid malignancies. So around about 25 different diseases, we can use this same process. And how we generate the labels is through several different ways. Up at the top, we feed in different, almost like dictionaries, where the first one would be CPT codes, so that's all the procedures. That would be everything from the drugs, IV drugs, through to surgeries, radiations, everything that we can label. So we extract all that data out, we label it up, and then we feed it back in with the labels. Also, ICD codes. So there is a few secondary or advanced stage codes, but they're not often used within a claim. They don't have to be used. But basically, if it's there, we want to utilize that. And also, we have pharmacy data that we use, and all of the precision medicines, they will all be oral in nature, so will be part of a pharmacy file or a prescription data. So we feed that in as well. So we have labeled all of that with particular stages as well. So we feed these 3 different things in, and then we have a series of rules after that. If something for a patient, for example, has been labeled with multiple different labels, we do have a hierarchy system on which one we will take. And at the end, we end up with all of our patient IDs, the stage of disease, and the date of diagnosis. So a patient can be identified as early stage on a particular date, then maybe at a later date be identified as advanced, or some can only be identified as advanced at diagnosis as well. So we get all of that. So together with taking our primary ICDs, our procedure codes, the secondary ICDs as well, our pharmacy, we're able to have this repeatable process to identify stage of disease. And what we've been able to do as well, because we do have access to all of the EHR data, or the electronic health records, and we've been able to validate all of our rules as well. So taking this a step further, in our rules we can make very simple rules, and that would be assigning a certain number of ICD codes to a particular disease. We can group particular CPT codes or procedures for biomarkers. We can do what we did before, which is add a lot of those things together, or we can get much more granular with our rules. And this is a project we worked on to get to a really granular metastatic lung cancer cohort. And this cohort, we had a lot of inclusion and exclusion criteria. This is just the overall flow diagram, and then we'll go into the Knime flow. But basically, we started off with all of our lung cancer patients, and then we had, there was 2 flows for this. So the patients could either have a primary ICD code for lung and a secondary ICD code for lung. So that was the first cohort. Then we built in an exclusion on surgical resections, and that exclusion was from 30 days before to 90 days after the first metastatic date, and that would be defined by the secondary ICD code. On the other side, we have another cohort of patients making this up. So it, again, would be by the primary ICD, but as I said, they don't always use secondary ICD codes. So in order to sort of mop all of those patients up, we used an IO chemo combination that would only be used in the metastatic setting. So that was here. Again, we had a number of exclusions. So we excluded on our surgical resection. Again, there was a time point of 90 days before to 120 days after. That would be based on the chemo IO date. And we also excluded on radiation and chemoradiation after completion of the chemo IO. A radiation block we would have defined here as at least 10 treatments within a 60-day period. And the chemoradiation block was 120 days prior to the chemo until 14 days after. So there was a lot of timings and, as I say, inclusions and exclusions here. So moving on, this is the flow that we built. And we're starting really with the 250,000 patients, so that would be our full lung cancer cohort. So here you can see in the yellow box this time, we have the identification of our primary ICDs and our secondary ICDs, and in the blue box at the top is our surgery. So at each time, the blue boxes are our exclusions. So, and underneath, I know it's very difficult to see, but the yellow, where it's highlighted yellow underneath, are the inclusions, and underneath, where it's blue, are the exclusions. So the primary and secondaries together are included, and then we bring it up and exclude for our surgeries. Likewise there, that's one side of it. On the other side, we were looking at our primary ICD codes, and then our chemo IOs. And then we excluded on radiation and chemoradiation at various time points, and then excluded on our surgeries as well. We were able to identify all of the metastatic dates, and again, if that happened if a patient, for example, was in both cohorts, we would take the minimum date that was there. So again, this will identify a list of patient IDs that are metastatic with their minimum metastatic date. So you can see we went from 250,000 patients to 50,000. And what was really good for us here was we worked really closely with our client in this case, and there was quite a lot of back and forth. We were able to use Knime, and it was really good to actually see those exclusions and check at each point of the pathway. Does this make clinical sense? Should these patients be included? Should we be extending certain time points? So at each point, we were able, with our clients, to go and show them snippets of the data and make sure that everything was clinically relevant for them. So just to summarize that then, in claims data, we can get to a particular disease. In this case, lung cancer, 250,000 patients. If our clients are interested in a metastatic setting, the journey in there is going to be diluted out by all of the noise of the other patients. We're not going to get a good signal of particular testing rates, because testing will not happen in every single patient. And we've generated our labels, and all of these labels, as I say, these are some of the ones for lung cancer. We have them across the board for all of our solid tumours. For instance, we can get to that early and advanced, which is what I've shown you today, but we do have things like newly diagnosed lines of therapy as well. And with these labels, we can start comboing them up. Our clients are very interested in getting to an advanced second line because that's where all of the new drug approvals will be. So they want to know what is the state of play in that patient cohort. So with the labeling system, we can do that and get there. So you can just see there where we've got our labels, we have 50,000 patients, and the data insights that we will get using this will be much more enhanced and valuable to our clients. So I will hand back over to Scott now to finalise. Oh.
Thanks, Wendy. So look, you can see the complexity in getting to one patient cohort for one client for one drug. We had to do this every single time for every drug that was getting released. There's hundreds of drugs in the pipeline at the minute, and we were really at a stage where, how are we going to scale this? You know, the analysts they used to sit down, the clinically animated folks used to sit down with the BAs, being the software BAs, and try and translate that into software code with their software engineering team. And I liked how Michael touched on that this morning. There's a massive translational layer. But the biggest insights we've ever found, the biggest wow factors we've ever had in our company didn't come from our software engineering team. It's not because they're not capable. It came from the clinically minded folks. The clinically minded folks that took the time to learn Knime and really embed that knowledge and utilize the underlying power of that data and the Knime platform to really extract that insight and get the patients on drugs, I guess. So a little bit about before and after Knime. The translational layer was tough. Again, there was really basic rule sets, had to do this every single time for every client. Wendy's team has got ahead of all of that now with their labeling system for hundreds of diseases and stages of diseases. If any client comes to us, they'll always want a different type of patient because our drug is targeting a different type of patient, and we're easily able to get the answers quite quickly now. So that's it, folks. I hope that was enough to not put you to sleep anyway. And thank you very much for listening.
Good. Thank you, Wendy. Thank you, Scott. Again, we have time, or we have actually lots of time for questions this time. Anyone from the audience? Oh, it's really light up here. Nobody? Good, then I have a question, actually. I mean, we've seen medium to even large-sized workflows in your presentation. How long does it take you to build those workflows?
Yeah, I think it's how long as a piece of string. So look, I think we've learned a lot along the way. We try to utilize the power of the database versus Knime at times. And it's really a trade-off then of how much SQL code can you put in versus how many nodes you use and things like that. So it really just depends. With the labeling system, advanced labeling system on board now, all of that logic, that rule set is pushed away back into the data engineering pipeline now. So we don't see that. We can still interact with it. But you imagine it really depends then if it's a new type of patient, a real custom type of patient that they're looking for, maybe like a misdiagnosis or something like that, then it could take anywhere from days to 2 weeks to complete. And that's really iterations with the pathologist, with the pharma company, sit down and really understanding the clinical ask.
Okay. We have one. Manuel, please.
Thanks, first of all, for this nice presentation. I'm wondering if the business processes that you introduced to us, if they are GXP relevant, and if they are, what effort did you take to make Knime GXP?
Sorry, could you repeat?
GXP.
GXP?
Yeah.
Can you?
With business practice stuff, pharma development, when you deal with FDA stuff, drug development, your systems usually need to be GXP compliant. That means they have to go through a huge validation process.
Yeah. So I guess we go through validation processes with each rule. That involves interaction with the pathologists and the clients, I guess. Internally, we are also going through ISO compliance as well, which involves a lot of our product and underlying assets within that as well. That is nearly wrapped up now, I guess. It's been going on for quite some time. It is quite painful, I guess, but I guess that's where we are in our journey at the moment.
Okay, thank you.
Any other questions from the audience? we have one question from our virtual participants. So he's really interested in these workflows that you have presented. In which stage of the diseases? Is there any way you can share the workflow, or is it like private, protected data? Obviously?
Normally proprietary. Like we can definitely discuss the processes that we've used, but yeah, the exact nuts and bolts normally are proprietary.
I figured that. Any other questions? Yeah, please.
As far as I understood, you used like existing rules to label your data. Have you ever tried to use like to put some kind of clustering algorithms to find new patterns, or is it just the existing rules?
So yeah, one of the issues you have is that with claims data itself, there is no, say, source truth as such. So yeah, we could go like an unsupervised method, but it's very difficult to know exactly who is advanced or early within that cohort. So it was much easier for us to actually build it from all of our rules and that clinical knowledge, and and that way we sort of know that we're along the right lines. We've obviously got our EHR data now, which is something new that we didn't have before. So potentially there is maybe scope now to do that, but before we couldn't really, because we didn't have that sort of, we didn't know how to call it correct. As such, but yeah, definitely something that I think we can move on to in the future.
Definitely, even it's kind of like pushing on a piece of string, going to a clinician with cluster A, B, C, and D, and asking them to, tell us what they are. If you can get a clinician to build that rule set themselves and use that clinical knowledge and bed that in, it's pulling on the string rather than pushing.
Okay.